Hi, my name is Manuel Cortes Mora, and I'm going to talk about micropropagation of Paulownia elongata via direct organogenesis. In this case, the name of Paulownia elongata is also Hiri tree, which is the origin of this tree is from Asia. It's a dicotyledon flowering plant. The family of this plant is Paulownia niaceaceae. The order is Lamilius. The importance of this plant or tree, it also that has the highest growing rate among trees. It also has the capacity to fixate 10 times more CO2 and also have the ability to adapt different types of soils with heavy metals or other contaminants. It is also viable for reforestation, and it is also used as a wood manufacturer and construction of industry. Our training partner is Wicklon, which is interested in the micropropagation of Kiri tree for production of quality wood, and also it also helps the Sustainable Development Goal 15 and 13, which is life on land and climate action. Our general objective is the germination of Paulownia seeds for further direct organogenesis in order to preserve the genetic identity and the characteristics of this species. The specific objectives of this is the germination of Paulownia elongata seeds in the MS media. In this case, to obtain healthy apical meristems as an explant. We also evaluate the induction of shoots through our proposed shoot induction media. Our hypothesis is that after 20 days from the seed culture, the germination rate is expected to accelerate and also less or non-contamination is expected as they were germinated in vitro. For the micropropagation, regarding the innovation for the shoot induction media, it is expected to observe more shoots per explant and larger shoots. Our proposals are treatment 1 and treatment 2. In this case, we also have an original treatment, which was from the paper, and our controls. To start this process, we first do the cleaning process, which the seeds were washed in a solution of 2% hypochlorite and 2 drops of 220 for 10 minutes with agitation. After this, they were rinsed in 3 times with sterile distilled water, and finally, they were transferred on the MS media, 10 seeds per flask. After this, we do the in vitro transfer of germinated seeds. After 16 days of this germination, 5 plants were extracted from the MS medium. They were transferred onto a 60 milliliters plastic cups with sterile substrate provided by the training partner, covered, labeled, and stored in the growing chamber at 25 degrees Celsius. After 28 days, 9 plants were also transferred onto a 250 milliliters plastic cups with sterile substrate, covered and labeled at 25 degrees Celsius. From where we want to use the micropropagation shoot induction media, we are going to use the explant as a apical meristem. These were excised with a scalpel from the sprout that had 2 nodes, and they were sealed with paraffin and placed in the incubator for 76 hours. After this, we are going to do 2 extracts, in this case from Paulownia shantung leaves and Gallithymia glauca leaves. Plants were rinsed with tap water and later washed with water and 5 drops of 220 in basin with gentle stirring. Then they were rinsed 3 times with the sterile distilled water, and finally they were placed in an aluminum foil to dry for 48 hours in the incubator at 40 degrees Celsius. For the extract, we are going to pulverize with a coffee mill. 1 gram of each was weighed with an analytical balance. Also, we are going to use 10 milliliters of methanol, and this is going to be added to the extract that was pulverized at 1 gram, or 1 gram of each one. This is going to be placed in a sonicator for 20 minutes, and next of that, we are going to centrifuge for 10 minutes at 6,000 RPM at 4 degrees Celsius. Then we are going to have the supernatant, and this is going to be retrieved using a syringe with a filter and covered with aluminum foil to be stored at 4 degrees. Then we are going to do the cytotoxicity bioassay. In this case, from the 2 extracts that I mentioned before. In this case, the concentration of the extract is going to be 25%, 50%, and 75% of extract After this, we are gonna do the experimental method of the cytotoxicity bioassay, where the cells were fixed into a 24 well plate with 70% ethanol and PBS. Then 400 microliters of violet crystal were added to each well. After this, the plate was stirred for 20 minutes. Partially, all the crystal violet was removed and 500 microliters of 70% ethanol were added and stirred for 15 minutes. Then the plate was rinsed and 200 microliters of each sample were transferred to a 96 well plate from testing the absorbance in the spectrophotometer. After this, we are gonna see the results of the in vitro germination. In this case, after 24 days, we see a 60% of germination rate, which was very, very good in this case. After the results of the first ex vitro transfer, unfortunately, it was not good. At this case, after 4 days in the incubator, 5 sprouts died, and only one survived, and that one was sealed with paraffin and to maintain the humidity. For the second ex vitro transfer, we used the one that survived and 9 sprouts more that were taken from the flask. They were placed in individual 250 milliliter plastic cups and sealed with paraffin to maintain the humidity. After this, we are gonna see the shoot induction media and the response variable of the control between 3 days and 7 days, in which case treatment 1 had better results in this case in 3 days and after 7 days also. And these are the apical meristems that were put in the MS media with our shoot induction media. And these are the explant where we excise these apical meristems that they are still growing. After this, we have the results of the cytotoxicity assay, which we see that are too high in this case. And in this case, we can say that it was an interference from the extract in the spectrophotometer test because this cytotoxicity assay needs to be decreased, no increasing. And the secondary metabolites of this paulownia, of this extract, is the anti-tumor, anti-cancer, and this is in the flowers, in the leaves, in the stem for Paulownia shantung. And for Galphemia glauca, we can see that it also have anti-cancer metabolites and gallic acid. For our statistical analysis, we can say that there is no difference between the mean of the length of shoots per treatment in the different levels of concentration. And our alternative hypothesis is that at least one of them, one mean of the length of shoot per treatment within the different levels is different, which means that at least one of the means is different of this. After this, we have that other null hypothesis, there is no difference between the mean of shoots per explant within the different levels of concentration of phytohormones, and at least one of the number of shoots per explant within the different levels of phytohormones is different. And for our ANOVA analysis, we see a p-value of 0.039 and 0.036, which is less than 0.05, with a confidence level of 96%, we can reject the null hypothesis and conclude there is a difference between the means of the number of shoots per explant and length of shoots within the different levels of concentration of phytohormones. To conclude, we could see that the in vitro demonstrate higher germination rates within few days in presence of activate charcoal on the germination in vitro. It also have, for the first ex vitro culture, 4 or 5 sprouts died for the absence of humidity. Hence, of the second transfer, the addition of paraffin increase the humidity in the plant and keep it viable for further observation must be made. For our shoot induction, treatment was exhibited to have better results than treatment 2, but we could say that treatment 1 has more shoots and treatment 2 also have more length. For the exit plants, the germination source plants showed growth and shoot development after the apical meristem was removed. Thank you for all.